Hello, everyone, and welcome to the Illumina Epigenetics experience. We will begin today's event with our first webinar, Clinical Utility of Whole Genome DNA Methylation Profiling as a Primary Molecular Diagnostic Assay for Central Nervous System Tumors, a prospective study and guidelines for clinical testing. I am Kaylee Bach of Labroots, and I'll be your moderator for this webinar, which is presented by Labroots and brought to you by Illumina. We encourage you to participate by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for after the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker, Dr. Matija Snuderl, the Director of Molecular Pathology at NYU Langone. For a complete biography on our speaker, please click their name in the presenter's window found on your screen. Dr. Snuderl, you may now begin your presentation.
Hello, everybody, and I would like to thank the organizers for giving me this opportunity to discuss our work in the whole genome DNA methylation profiling and to present the clinical utility and first clinical guidelines for Hello, everybody, and I would like to thank the organizers for giving me this opportunity to discuss our work in the whole genome DNA methylation profiling and to present the clinical utility and first clinical guidelines for DNA methylation analysis for CNS tumors. My name is Matija Snuderl, and I'm Director of Molecular Pathology at NYU Grossman School of Medicine, NYU Langone Health. So, patients with brain tumors face many difficult choices, and NYU Brain and Spine Tumor Center is comprehensive clinical unit which takes care of all the patient's needs for therapeutic, diagnostic, as well as research questions. We have comprehensive services that take care of patients from the beginning to the end of their journey, and DNA methylation testing is an important part of it. Treatment of patients with brain tumors is complex, and it starts with a maximal surgical resection followed by either observation or, if additional therapy is required, radiation or chemotherapy, for example, temozolomide for gliomas. But one very important point in patients' journey between a resection and any kind of a therapy or follow-up is diagnosis, and that will be the focus of my talk today. It is important to keep in mind that while brain tumors are only 2% of all cancers, they represent an important number of lives lost due to cancer. Brain tumors are cancers with the fourth highest number of lives lost. They're the second most common cancer among children between 0 and 14 years, and the leading cause of cancer-related deaths in children. So despite a low prevalence, they have oversized impact on quality of life and patients' outcomes. The problem of diagnostics of brain tumors is that they represent an incredibly diverse group of diseases, both from the clinical as well as the molecular standpoint. We currently have more than 120 different types of primary brain tumors and across the brain and spinal cord that are driven by a diverse set of molecular drivers that include mutations, gene fusions, copy number aberrations, including amplifications and deletions, and some of these molecular drivers still remain unknown. So what are molecular alterations in cancer? Well, we can talk about mutations in the DNA, overexpression or underexpression or downregulation of different genes through RNA, and gene fusions, or dysregulation of proteins. But what we are particularly interested in today is epigenetic changes, methylation of DNA. And NYU Molecular Pathology has developed a very comprehensive molecular diagnostic portfolio, which includes NYU Langone Genome Pack, which is currently the largest FDA-cleared NGS assays for somatic mutations in cancer. We were the first CLIA-certified laboratory to validate and obtain regulatory approval for NYU whole genome DNA methylation, and we analyze gene fusions through a comprehensive NYU Fusion Seeker platform. You may ask yourself, why do we want to deal with DNA methylation? Well, there is something that I call BRAF600E paradox, which is a mutation commonly present in a variety of human tumors, from most malignant tumors such as malignant melanoma to some of the most benign tumors such as pilocytic astrocytomas, and a large number of tumors in between. So what is important to keep in mind is that mutations simply do not tell the full story of a disease. It is really important to keep in mind that if you were just to analyze mutations, you would not get a full picture of what is driving the tumor and how it's going to behave. DNA methylation is a chemical bond of DNA to a methyl CH3 group. DNA is methylated in tumors all along the genome. And what is important is that the pattern of sites where DNA is methylated truly represents a fingerprint that is passed from a cell of origin of the tumor all the way down to the later clones. And DNA methylation can tell us both about the type of a tumor as well as an aggressiveness, and it can help us identify the molecular drivers of that tumor. How do we do it? Profiling of tumor methylation is performed using Illumina DNA methylation EPIC B-chip array, although there are many other methods through which you can analyze DNA methylation, for example, methylation sequencing. The advantage of the array is that it is an incredibly robust assay, and the results are comparable between different labs. CNS tumor patients can be profiled with this approach, and the advantage is that it's really a pan-tumor approach, so it doesn't really matter what kind of a tumor the patient has, and DNA methylation B-chip is a high throughput, which utilizes these beads on which methylated and unmethylated DNA binds to deliver the image of from the array that we can decode to establish methylation profiles. Another advantage of DNA methylation array is that it can replace several other assays that would otherwise have to be analyzed through a different molecular analysis. We can predict IDH mutation status, ATRX mutation status. We can analyze chromosome 1p90q deletion or analyze third promoter status, all through a single assay and the bioinformatic pipeline that we developed a few years ago in collaboration with the Solomon Lab called UDD. In addition, as you can see here, we can use DNA methylation data to derive a copy number plot to look for copy number aberrations, including amplifications, deletions, and overall copy number status. We can further look into the promoters of genes we're interested in, such as MGMT in brain tumors, but also in other cancers, MLH1, so we can identify promoter methylation status. So what we really want to focus on is how do we define brain tumors molecularly rather than histologically. So in this slide, you can see how not that long ago, less than 10 years ago, we used to classify all brain tumors using histopathological slides alone, as like a glioblastoma grade 4, medulloblastoma grade 4, and ependymoma. But today, based on a large landscape studies using sequencing methods and DNA methylation, we now know that each of these histological entities is composed of multiple molecular subtypes that have a distinct clinical, pathological, and outcome features. So now it is not sufficient to call it medulloblastoma grade 4. We now know that these tumors can be subclassified, and it's important to subclassify them, for example, as a sonic hedgehog subtype. Similarly, high-grade gliomas can be further subclassified, or ependymomas. So precision for molecular diagnosis really allows for better prognosis assessment and treatment options. As an example, I would like to show you two patients here from NYU, which both have the same posterior fossa brain tumors highlighted here in the MRI images above. And you can see, using traditional diagnostic with histological images, they both get the same diagnosis, so a WHO grade 4 medulloblastoma. But when you perform molecular analysis, we know that one of these patients will have a Wnt subtype, the other one has a group 3 subtypes. And it's pretty clear that if one child has almost 100% chance of survival and the other child has only 30% chance of survival, these are not the same diseases clinically. These two children have a different risk of recurrence, long-term survival. They will need different therapy, long-term management, and perhaps one day precision therapy. So a few years ago, a large effort has started to develop a pan-CNS tumor map of brain tumors using DNA methylation. And these are published data from landmark paper published in Nature in 2018. It's a little bit of a complicated picture, but let me show you here. Each of these rectangles correspond to specific types of brain tumors as defined by WHO or equivalent. All these tumors are profiled using DNA methylation, and you can see here on this diagonal chromosome-like band that all these rectangles, all these different colors correspond to different tumors, and you can zoom in here into a triangle of ependymal tumors that is then further substratified in additional molecular subtypes of ependymoma. So how does it really work? We take a tumor of interest, we are agnostic to it, we run it on an Illumina DNA methylation array. It is compared to a reference cohort of now more than 5,000 tumors and more than 110 entities, and we derive a result of what the tumor is. What is important to emphasize is that all of these classes have been defined with additional orthogonal approaches, either morphologically, by sequencing, copy number analysis, expression profiling, or others. So we have a good reference set to which each of the unknown tumors is compared. So just to give you an example how this technology works, this is a patient who is a 17-year-old female who presented with seizures. Imaging showed enhancing tumor. She underwent subtotal resection and with a pathology called malignant tumor. You can see here MRI and histological and immunohistochemical results. Tumor that had both glial as well as neuronal expression as shown by GFAP and synaptophysin. And you don't have to be an oncologist or pathologist to agree the diagnosis of a malignant tumor is really not helpful for clinical management. So the patient received 2 expert consultations. One of them called it the GBM, WHO grade 4, with the tumor board recommendation for radiation and glioma-type chemotherapy, including temozolomide, while the other expert classified it as a primitive neuroectodermal tumor, WHO grade 4, with the recommendation of a craniospinal radiation and PNET chemotherapy. So you can see these are 2 vastly different diagnoses with a completely different management options. So patients came to NYU for a third opinion because of these conflicting diagnoses and clinical recommendations, and we performed DNA methylation to classify her brain tumor, and based on DNA methylation, we classified this tumor as a pleomorphic xanthoastrocytoma WHO grade 2. So what are the implications? We now have a result that says that the correct diagnosis is a pleomorphic xanthoastrocytoma, not a glioblastoma, not a PNET. Correct grade, WHO grade 2, not 4, and she underwent gross total resection, not radiation or chemotherapy, and she's been disease-free. So you can see a significant impact on the quality of life and outcome of this patient using DNA methylation profiling. I want to show you another case with a 7-year-old boy who presented originally with a progressive headache and nausea, was admitted to Columbia, and MRI showed obstructive hydrocephalus because there's a 4th ventricular mass. MRI showed multiple leptomeningeal metastases. Patient went gross total resection with a diagnosis of medulloblastoma and was then treated at NYU on a CCG 99701 protocol and remained in continuous remission after that. 12 years later, at age 19, incidental finding of surveillance MRI showed a new nodule enhancing right-sided brachium pontis lesion, which grew over a 2 months period, and it was concerning, and biopsy was performed. When we look at this tissue, you can see here in histology a tumor composed of relatively uniform small round blue cells, reasonably negative for GFAP with some expression of synaptophysin that was unclear whether in cells in the background, with a retained INI1, no positive for T27, but extremely high proliferation index. So a preliminary histology was suggestive and compatible with a recurrent medulloblastoma. But when we performed DNA methylation profiling, to our surprise, we showed that the tumor actually classified best as a pediatric diffuse high-grade glioma with an RTK1 subtype subclass novel. So you can see a complete switch from the diagnosis into a glioblastoma with multiple copy number aberrations suggestive of radiation-induced GBM. In addition, as I showed you before, the advantage of DNA methylation is that we can see copy number plots, and we can also analyze MGMT promoter status with DNA methylation. So who is right, who is wrong? You may ask, well, how do we know that DNA methylation is correct? We perform NYU Langone Genome Pack, which is our FDA cleared tumor normal 607 gene panel, and the results were compatible with the glioblastoma. You can see amplifications of PDGFR alpha, CHIK2, and homozygous deletion of CDKN2A with a several mutation. So this mutational copy number status is really compatible with glioblastoma. So results were glioblastoma, not recurrent medulloblastoma. The patient underwent correct treatment for glioblastoma, but it also raises important epidemiological questions. What fraction of recurrent medulloblastomas are actually secondary GBMs? And what is the survival analysis and assessment of therapeutic efficacy of tumors that recur? It is as important for design of future clinical trials. If this patient was diagnosed as a recurrent medulloblastoma and enrolled in such a trial, it would not be a good candidate for it. And it also brings a question of potentially de-escalating primary tumor treatment to decrease the risk of secondary tumors. Misdiagnosis has a significant impact on clinical trials. This was the reanalysis of ACNS 0332 trial, which was a phase 3 4-arm prospective COG trial, which was focused on PNET and pineoblastoma. The trial was, I believe, closed because it was deemed to be unsuccessful, and it basically failed. But when authors went back and they reanalyzed all the patients enrolled on this trial with a DNA methylation, they showed, to a huge surprise of everybody, that more than 70% of these so-called PNET cases were actually represented by molecular-defined entities that were not intentionally, they were not supposed to be included into this clinical trial. So 70% of these patients were never supposed to miss trial, and if you remove them, the trial would actually be a success. So that shows you that can have a significant impact on clinical trials if you enroll a large number of patients that should have never been included in this clinical trial. So the question is, and that's something we asked ourselves many years ago, is how frequently this actually happens. So the problem is that many of the early DNA methylation studies focused on challenging so-called hard-to-diagnose tumors. There was a significant selection bias. Studies were retrospective, and there were no clinical guidelines for pathologists or oncologists who would like to use this test in clinical practice. And we focused on analyzing data prospectively, and this is a recently published study on a clinical utility based on this prospective study. So how do we design this study? Well, first, we aim to establish the clinical utility across all CNS tumors to answer definitively for which brain tumors DNA methylation is useful. We designed as a prospective biomarker study, included almost 2,000 primary CNS tumors diagnosed at NYU and sent to NYU between 2014 and 2022, and included all newly diagnosed brain tumors tested by DNA methylation. So there was no selection bias for difficult cases or select cases. We pretty much profiled every tumor that had sufficient amount of tissue, and then we performed orthogonal DNA and RNA NGS studies for confirmation and to resolve discrepancies to establish who was right, histology or DNA methylation. So in panel A, you can see the design of the study, and then let me walk you through the panels below. So in panel B, you see a total breakdown of our cohort. You can see that more than 80% of studies had a WHO diagnosis, but 70% of tumors included in this study carried some sort of a descriptive diagnosis, and we're going to talk about it more in detail. In panel C, it is important to realize that out of the tumors that carry the WHO diagnosis, almost 75% of all these tumors, DNA methylation agreed with the original diagnosis, but in 14% of cases, 225 cases, there was a difference between the original pathology and DNA methylation. In addition, 7% of cases, there was an agreement between histology and DNA methylation, but DNA methylation provided additional prognostic value. For example, diagnosis of medulloblastoma further substratified into prognostically important subtype. In 5% of cases, there was no match, and DNA methylation was not able to find a match for the tumor. In tumors that carry descriptive diagnoses, this is one of the important parts, is that more than 85% of cases were resolved using DNA methylation. We're going to talk about it more in detail shortly, and it's about 14% of cases that DNA methylation was not able to match to any existing class. What are the reasons for the no match? The most common reason is the low tumor cell content. DNA methylation requires a relatively high percentage of tumor cells. Another common reason was a poor DNA quality. Some of the cases probably represent unknown or novel tumor types. To surprise of many of us, we keep discovering and publishing and describing new brain types of brain tumors. Another possibility is of a novel or rare molecular drivers, and also for some of the tumors that were believed to be primary CNS tumors, we cannot rule out that some of them may be a metastasis coming from the unknown primary. When we look at the tumors that carried WHO diagnoses such as glioblastoma, ependymoma, oligodendroglioma, or others, to our somewhat surprise, we noticed that even in the tumors that carried a very clear WHO diagnosis, there was still a significant percentage of patients, like I'm highlighting here, that carried DNA methylation reclassified. And that's important because we often get asked, well, which tumors should I profile? And if I know what it is, does it make any reason to perform DNA methylation? But as Mark Twain said, it isn't what you don't know that gets you in trouble, it's what you know for sure that it just isn't so. So even tumors that pathologists think that they know how to diagnose them, there can be a significant percentage of diagnostic errors as shown here. So this shows us that even in tumors with well-established WHO diagnosis, DNA methylation can be incredibly helpful. Another point is that in our cohort of almost 2,000 tumors, there was a 319 tumors, 17%, that were diagnosed descriptively and carried 195 different descriptive diagnoses. So that's a huge number, and we really pulled all different diagnostic terms, being a low-grade glioma, NOS, to high-grade tumors, C-node, neuroepithelial tumor, and many, many other things. And descriptive diagnoses such as this are a plague both of pathologists as well as an oncologist because they fail to deliver a clear message what the tumor is and how it should be treated. And here you can see that methylation was able to resolve 86% of all these cases, and you can see we're able to reclassify 195 different descriptive diagnoses into a limited number of 50 or something methylation classes as shown in these panels. So it really narrows down the number of diagnostic terms, gives precise diagnosis, and helps clinicians with the clinical management. Another important part that I would like to mention is many of the descriptive diagnoses try to indicate to a certain extent whether the tumor is a high grade or low grade, even if a pathologist cannot really definitively diagnose it. So the category could be, for example, high grade neuroepithelial tumor, which conveys to a clinician a message that the patient should be treated as a high grade, while the low grade tumors, if gross totally resected, can often be selected to just be observed. And that's a significant message that pathologists are giving to the clinician. What you can see here from this plot is that more than 20% of patients with a high-grade diagnosis actually end up being reclassified into a low-grade methylation class, and approximately 25% of low-grade descriptive tumors end up being reclassified into a high-grade methylation class. So you can see that we are able to not only decrease the number of diagnoses from 195 to approximately 50 DNA methylation classes and provide definitive diagnosis for clinical management in over 80% of cases, but we're actually able to change the grading of tumors and therefore influence the management in approximately 20% of cases, which is very significant. Based on this prospective study of 1,900 CNS tumors, we crafted the guidelines that we hope will help our colleagues, both on oncology and neuropathology side, to decide how to best utilize DNA methylation. We have stratified brain tumors into 3 categories: high yield, intermediate yield, and low yield. So the high yield tumors are tumors which are defined by DNA methylation signatures as described in WHO. And we believe that high yield are all CNS tumors with descriptive diagnosis. Considering that DNA methylation can resolve more than 80% of descriptively signed out cases, there is simply no reason to not pursue DNA methylation in tumors with descriptive diagnosis. High yield cases are also all the tumors where there's a high chance of diagnostic error in the absence of other molecular studies, and all tumors with inconclusive or contradictory immunohistochemical or molecular results, and tumors where there's established impact of clinical management based on prognosis. Intermediate yield are tumors where DNA methylation could triage further molecular testing. For example, if there's a limited amount of material and you cannot perform all the NGS studies, DNA and RNA, DNA methylation can help you stratify for that. Tumors with moderate chance of diagnostic error in the absence of molecular studies. And in addition to that, all the tumors in which more than 10 immunohistochemical stains would be needed or many molecular tests would be required for diagnosis, which helps you with the tissue preservation and cost efficiency. We often see tumors where 40 or 50 immunohistochemical stains are performed and no diagnosis can be reached. And it just takes one DNA methylation test to get to that final diagnosis. There are low-yield tumors for which currently DNA methylation does not have a specific use, which are tumors where other molecular tests have sufficiently established molecular drivers in tumor classification, tumors that do not have currently prognostic value based on molecular subclassification, and then tumors where there's a very low chance of diagnostic error when using recommended techniques, according to WHO, for example, schwannomas and pituitary adenomas. Although the field is changing quickly, so some of them may become important from the prognostic standpoint. I want to make a little segue and just show you that combining a power of different methylation classifiers and building a whole body epigenetic map of cancer is not that far from the future. This is a patient who was a 22-year-old female with a diagnosis of pineoblastoma approximately a year before she came to our institution. She was a status post CSI and adjuvant chemotherapy per COG medroblastoma protocol and presented with a rapidly recurrent disease with local recurrence. And you can see there is a midline tumor here on the MRI, and there's a small round blue cell tumor. So the diagnosis of pineoblastoma is not unreasonable. But when we performed DNA methylation profiling, there was no match by CNS brain tumor classifier. So we are pretty convinced that this is actually not a brain tumor, at least not an entity present in the classifier. And when we perform additional studies, and it's a published paper, we saw that the brain tumor actually clustered with alveolar rhabdomyosarcoma, and RNA sequencing confirmed the diagnostic fusion as shown here in the panel C. And here you can see in the heat map, the tumor beautifully clusters in the middle of the alveolar rhabdomyosarcoma cluster. So it shows you that we can identify tumors that are not typically considered to be brain tumors, at least excluding all the non-brain tumor entities. And when we follow up with additional immunohistochemistry in the light of this diagnosis, you can see the tumor light up very brightly with both myogenic and desmin, so it ended up being a re-diagnosed or correctly diagnosed as a rhabdomyosarcoma with PAX3 and COA2 fusion. A sarcoma classifier has also been published and is currently being used in clinical practice by our institution and many others. So whenever we see a tumor that doesn't classify well on the brain tumor classifier, we always rerun it to a sarcoma classifier exactly for these purposes. And this is just the beginning of a much more exciting development that, I believe that DNA methylation and AI-based classifiers will really become a dominant diagnostic method in cancer, not only for brain tumors and sarcomas, as I've shown you, but there's also a sinonasal tumor. There are other work on hematological malignancies using DNA methylation, as well as the salivary gland tumors, kidney tumors, and a couple of other examples of molecular subtyping for prognostics or breast cancers or DNA methylation-based subgrouping of lung adenocarcinomas. So in conclusions, I hope I showed you that molecular pathology area of precision diagnostics and in-depth analysis can allow for more accurate stratification of cancer patients for clinical management. There's a significant inter-observer variability based on histology alone that leads to discordant diagnosis in about 12% of academic institutions and about 25% of community hospitals. DNA methylation is currently the only pan-CNS tumor assay that incorporates detection of all other biomarkers, decreasing the cost, turnaround time, and tissue waste associated with sequential molecular testing and diagnostic errors. DNA methylation is also a diagnostic tool that really improves diagnostic accuracy, decreases inconclusive diagnoses, and provides prognostic subclassification that is only going to further increase in importance. And then we provided you with the guidelines based on unbiased prospective cohort and recommending stratification of CNS tumors based on diagnostic yield and possibility of diagnostic errors. This work would not be possible without many people working on it. I would like to acknowledge NYU Molecular Pathology Laboratory, NYU Brain and Spine Tumor Center, our collaborators from the KFZ who really pioneered this work, and multiple funding sources, both foundations and NIH, that helped over the years support this work. And with that, I would like to thank you for your attention, and I'll be happy to take any questions. Thank you.
All right. Well, thank you, Dr. Snuderl, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question that you'd like to ask, please do so now. We'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here is, does patient ethnicity play a role in the DNA methylation classification?
Thank you, Kaylee. Can you hear me well? Wonderful. Yeah, so thank you again to the organizers for giving me the opportunity to present our work on DNA methylation. It's been an incredible journey and a great support of many people. So that is a great question. And I think you really start with the hardest question because we are just scratching the surface. There's so many things that influence epigenetics, and our original idea that every tumor is going to have a very discrete and straightforward epigenetic signature turned out to be somewhat naive. And it's as we work on more tumors and we have a larger and larger data sets and larger populations, we see that there are many things that shape epigenetic signature, and ethnicity turns out to be one of them. And we have seen that there are definitely differences between different populations. However, that will require much larger populational studies to be able to match the ethnicity to different signatures. But the simple answer is yes, it does. It helps us to identify which cancers are more prevalent in different populations. We recently submitted a paper on that topic outlining DNA methylation signatures in different ethnic backgrounds. And I think as we expand, we're going to be able to also figure out which of the molecular subtypes of tumors are more associated with unique ethnic backgrounds.
Great, thank you. Now I have another question for you here. What do you see on the horizon for this epigenetic approach?
Yeah, so horizon is really quickly coming closer. I showed you how we utilize DNA methylation for brain tumors. That's already clinical reality. It's being maybe somewhat slowly but readily incorporated into the guidelines. We presented our laboratory guidelines that we coined at NYU, and we think they're very practical for people who are thinking how to implement in a clinical practice. There's been some updates in the NCCN guidelines and a lot of talk in the CAP and USCAP, how to best implement DNA methylation, and both Association for Molecular Pathology and CGC are working on the positions and white papers how to implement them. So brain tumor is a given. I showed you sarcomas. That's sort of like a next big one that is coming in, but there are many other classifiers on the horizon. For example, NCI group has developed a beautiful classifier for hematological malignancies. We are working jointly on a kidney cancer classifier, and our lab has recently finished work on metastatic tumors to be able to use DNA methylation for classifications. So I think horizon is that DNA methylation will become one of the dominant diagnostic techniques in molecular pathology.
Great, thank you. All right. Now, what is the typical turnaround time to do DNA methylation for CNS tumor classification?
It depends where you are. I would say in many laboratories, there is, they don't have a volume that would justify running methylation array every week. One of the disadvantages, it's a high throughput test. So it doesn't, you can't really do one off or two off, right? Each chip has 8 slots, which means you can run 8 patients at a time, and you really cannot save the empty slots, right? We are fortunate, and we are a big reference center, and we really profile both brain tumors, sarcomas, and other cancers. So we run it every time. So we usually quote 7 to 10 business days. So if you haven't incorporated nicely in your workflow, it's about 7 to 10 business days.
Great, thank you. All right, next question here. This has a few parts to it. This question asks, does microarray methylation have SNP probes? If not, is there any plan to include SNP probe in the future? And do you think it is useful to have SNP probe at all to detect LOH?
So I think the simple answer is it does. It has some SNP probes for genotyping. Caveat to that is it doesn't have many. So you cannot use it in lieu over genotyping. It's not really, it's good enough to make sure that the samples belong to the right patient. But for example, there have been some packages that suggest that you can use methylation array also for ethnicity annotation, back to our first question. And it turns out it's not as, obviously it doesn't have that many SNPs, so it's not as informative as the others. So the answer is it does. The part B is not many. Would it help if it had more? It could help, but I would say it would take in the expense of some of the more important things that methylation array covers. It ends up being a matter of real estate that you have on the chip. So it's a bit of a trade-off.
Great, thank you. All right. Now, are there any malignancies in which there is no indication for DNA methylation studies?
I think that's a loaded question. It's like asking, is there any cancer there is no reason to investigate epigenetics? I think I would parse it a little bit differently. I think there are tumors, as we've shown in our guidelines, where currently a role of DNA methylation classifier is limited. A good example of that is a schwannoma. Schwannoma, it's easy to diagnose, hard to make a diagnosis, and very little, if any, prognostic significance, right? That may change in the future, right? If there is a therapy that treats schwannoma, maybe we'll be able to come up with a prognostic biomarker, predictive biomarker from DNA methylation. So I would say there are tumors where utilization of DNA methylation classifier is limited. I would say from the sampling perspective, tumors that have a very low tumor percentage are also not very helpful, just because the cancer epigenetics gets diluted by normal cells epigenetics. So I'd say poor samples with a low tumor cell content and samples where the currently DNA methylation does not really have a clear diagnostic or prognostic role would be of lesser importance. But that may change in the future.
Great, thank you. All right, next question here. Is sequencing a better or more comprehensive approach? And is 5-HMC important?
The sequencing is more comprehensive because array is limited in the terms of a number of sites it can investigate. So if you have the money and you have the samples, sequencing will be more comprehensive, but it also requires more comprehensive pipeline for analysis. I would like to point out that in a clinical setting, you're working with a formalin-fixed paraffin-embedded tissue, and we have learned through a very painful process that current bisulfite sequencing techniques are not very good for formalin-fixed paraffin-embedded tissue. So for clinical samples that you're working with an FFPE block, the advantage of array is that it's incredibly robust because it works with the smaller fragments of DNA that bind really well to the bead chip, right? So if you're working in FFPE samples, your options for sequencing are limited. But if you have a good amount of money and you have very fresh, very high quality DNA, snap frozen, then sequencing will give you significantly more real estate coverage.
Great. We're getting some great questions in from our audience, and we have some time left, so we have time for a few more. This next question is another two-part question for you. Anne asks, what are the biggest bottlenecks to make this array-based technology more widely adopted across the world from your perspective? And how can Illumina help to achieve this?
Yeah, so, we were fortunate to be at this journey from the beginning, and it wouldn't, I need to say, it would not have happened without an incredible support from philanthropy that believed in our goal to improve diagnostics. Unfortunately, we were not fortunate with the NIH funding. Every single one of my grants focused on development of DNA methylation for diagnosis was readily rejected as not being mechanistic enough. But the great philanthropic support from many of our donors really helped us propel this forward. So it actually boils down to the money, right? So we want this technique to become a standard of care because it really prevents diagnostic errors. Prevents diagnostic errors and improves clinical management, right? But to make it a standard diagnostic test, there has to be a standard workflow procedure that it doesn't limit the assay only to the labs such as ours that have a philanthropic support, right? And for having a laboratory test that is regular, it has to have a billing code. There has to be a process how labs can get paid for the work that they do, and it has to be accepted both by payers and the providers that this is a useful laboratory technique. And in terms of and there's a significant effort that I've been involved with in making sure that this assay, which is a combination of a wet lab work and machine learning, gets its own appropriate CPT code, so labs can actually bill for it, and there can be negotiations with the payers to get it paid. Otherwise, we will limit this assay to a handful of ivory towers on the east or west coast, where they can afford doing it with a philanthropy support. But that's not a long-term solution, and it takes a lot of patience out of equation. Patients with brain tumors live everywhere. This should not be a boutique test reserved for only selected populations of patients that live close enough to NYU or other fortunate institution. This really has to be a general lab test.
Great, thank you. All right, this next question here asks, is your reference classifier available publicly, or do you have plans to make it available for the public?
So this is actually, I have to say, this was developed at Heidelberg, the German Cancer Center, and the Nature 2018 paper has the original classifier data set publicly available, so anybody can download it. There's an effort on publishing and making available a data set for the updated version 12, which has an additional 25 types of tumors that will be made available as well for non-commercial and research purposes.
Great, thank you. All right, next question. What are other tumor applications utilize DNA methylation at NYU?
Clinically, currently, brain tumors, we are validating sarcomas, and the next things we'll see, we'll probably launch the heme and probably kidney.
Great. All right, and it looks like we have time for one more question here, so we'll wrap up after this one. This question asks, what library preps have you tried for DNA methylation?
Array is not a sequencing technique, so it doesn't have library preparations, has a different workflow. I would refer to Illumina website, have a very detailed protocol, and we have published a very detailed hands-on protocol, Snuderl Serrano a few years ago. That covers very detailed protocol, how to perform it. In terms of library prep protocols for sequencing, we tried the method capture TrueSeq, which is a pretty reliable method.
Thank you, Dr. Snuderl. We have time for one more quick question that came in, so we'll ask this one before we wrap up. This question asks, do you use Bethesda NCI classifier as well?
We have a very close collaboration with the NCI group, and it's a collaborative effort between both groups. Each of them has some advantages and disadvantages, and all labs use all of them. We have clinically validated German classifier, which has a New York State approval. We exchange samples with NCI and we consult on difficult cases, but clinically validated, we use the German classifier, which is very robust. And we have shown it with the Prospective study, which is sort of like a benchmark, that it performs well. So clinically, the German classifier, but very close collaboration with NCI and exchanging challenging cases. Very nice collaborative work, working together on being able to address complex cases.
Great. Well, thank you, Dr. Snuderl. Do you have any final comments for our audience before we close?
I mean, it's been a real pleasure working on this. It's nice to see how we were able to translate, this technology really from basic research all the way into the clinical space, and now seeing it basically transforming WHO classification of brain tumors and the clinical guidelines has been a real privilege, and it really shows the impact of translational research on clinical care. Thank you.
Well, we want to thank you again, Dr. Matija Snuderl, for your time today and your important research. We would also like to thank Labroots and the Illumina team for making this event happen. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webinar will be available for on-demand viewing after the live event. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. We'd also like to encourage you to continue to explore the virtual event, and don't forget to join us in the networking lounge to chat with the Illumina product management team at 12 p.m. Eastern Time. Until next time, take care, everyone. Goodbye.